I'm going to introduce Dr. Krishnendu Guin, who is a newly minted postdoc in Tom Mastelli's lab, but we're hearing his wonderful work from his his thesis work in Kostov Sanyal's lab from the Jawaharlal Nehru Center for Advanced Scientific Research. And there, Krishnendu did a lot of beautiful work on centromeric interactions and evolution in different yeasts. In fact, his most recent paper was recommended by the faculty of 1000, which is a fantastic honor. So I'm really excited to hear your work today. And without further ado, I will give the floor to Krishnendu. Thank you so much for being here.
That's great. Thank you so much, Christina. And I really appreciate you guys, what you are doing here. And I'm really happy to be here to present our work in front of you in the Fragile Nucleosome seminar series. So I'll just start. So the work, as you said, the work that was done during my PhD, this was one of the projects that we pursued during the PhD training. But now that I look back to the story, it seems like a story to me. And what I'm going to tell you today is a very old story. And this is a story of centromere evolution that happened about 39 million years ago. So let me begin by introducing you to the centromere. I'm sure you guys know about centromeres, but still, I'd like to begin by defining the centromere. And if you know that the centromeres are the genomic locus, which is the DNA sequence that specifically bind to the kinetochore proteins and assemble the kinetochore microtubule interface that allow the chromosomes to segregate during the cell division and maintains the genomic integrity during the cell division and in the daughter cells. So the cell requires to maintain this genomic stability, and the centromere plays a key role in maintaining this genomic stability in this process. But the interesting thing is that when we are looking at the centromeres across different species, what we find is that there is no magic sequence that define that this is the kinetochore assembly site and this is where the microtubules would bind. So if you look at this figure here, this shows you that the centromere DNA sequences across different species are highly different in terms of their length, their repeat content, their structures. They're very, very different from each other. So this is something which is known as centromere paradox in the field, and people are trying to understand what is the contribution of DNA sequences in establishing the centromere identity. So this is also same for the fungal kingdom, and today about 50 fungal species have been studied for the centromere structure and functional properties, and this is something we reviewed last year. And as you can see here in this figure, the centromeres in fungal species are widely diverse. For example, you see the single nucleosome length centromeres in Saccharomyces cerevisiae, which is 125 base pair long. And then you have centromeres, which are longer than 100 kilobase pair, and so on and so forth. So when I started my PhD, I was really fascinated with this situation that the conserved biological process of chromosome segregation is controlled by an element which is not conserved by itself. So this is the problem that I eventually had worked on. And when I joined the lab, my senior Gautam was working on the identification of centromere in Candida tropicalis. And what we found is that the centromeres in Candida tropicalis have inverted repeat associated centromeres, and the centromeres are highly homogenized sequences across all 7 centromeres, as you can see in this figure. And we are talking about more than 80% sequence identity across the centromeres. So they are really homogenized. Now, as you can see here in this figure, we looked at the localization of different kinetochore proteins, and in different stages of cell cycle, you see that per nucleus, you see one puncta in the interface, and in mitosis, you see them segregated. So this tells you that the centromeres, 7 centromeres in Candida tropicalis, are clustered in one spot, which is below the resolution of light microscopy. So this is something which is referred to as Ravel-like organization, in which you have the centromeres clustered near the SPB, and this is at the periphery of the genomic structure. So this is something which is also conserved in other fungal species. So there is no surprise here, but identification of the inverted repeat associated homogenized centromeres was something that we didn't expect to find. And this presented us with an opportunity to ask a question: what happened during the evolution that these two guys, Candida albicans and Candida dublineensis, they have really unique centromere sequences. What it means is that all the 8 centromeres of Candida albicans and dublineensis are unique DNA sequences. There is no homology whatsoever. But you look at Candida tropicalis, 7 centromeres of Candida tropicalis share almost 80% sequence identity, and all of them have inverted repeat associated structure. So we asked this question, that what happened during this evolution that the inverted repeat associated structures were lost, and you have unique centromeres in Candida albicans. So this is the question that we wanted to address, and we were thinking what happened. So this is the hypothesis that we came up with, because we knew that the centromeres of Candida tropicalis and Candida albicans are clustered. So we asked that, is it possible that the last common ancestor of these two species, they also had the centromeres clustered together? And if the centromeres are homogenized inverted repeat associated sequence in the last common ancestor, if there's a break in one of the centromere, they can go through translocation and lose the inverted repeat associated structure to produce the unique centromeres, which we see today in Candida albicans. So we wanted to test this hypothesis, and to test this hypothesis, we needed to assemble the genome of Candida tropicalis, which was not available when we were trying to address this question. So we started by assembling the genome of Candida tropicalis, and we employed various different sequencing techniques, and we also validated the genome assembly with SafeGel karyotyping, and we assembled the genome of Candida tropicalis in 7 gapless chromosomes. So having this gapless genome assembly allowed us to do 2 things. First thing is that we looked at the genome organization in Candida tropicalis, and this is a 3C sequence, 3C heat map. And you see that the diagonal is the most enriched contacts. That means the neighbors of the genomic regions interact with each other with highest affinity. And also you pick up some of the spots across these matrix that tells you the centromeres, which are the notch in the chromosomes aligned on the axis of this plot, tells you that the centromeres are interacting with the other centromeres. And you see that interaction for all the combinations possible for 7 chromosomes that is present in Candida tropicalis genome. So this is expected because previously we have seen that the centromeres are clustered in tropically. So this is what we expected. But we also found something new, that the centromeres, which are clustered together, are not really interacting with the other regions of the genome. So if you walk away from the centromere towards the chromosomal arm, you see the interactions dropping here, which produce a cross-like pattern. And that tells you that centromeres are excluded from interacting with the rest of the genome. So, and also if you look at the telomeres, you see the telomeres have some amount of interactions which are enriched compared to the rest of the genome. So based on this information, if you draw a cartoon, it will look like this, that the centromeres are clustered together, and the arms of the chromosome are extruded outwards, and the telomeres are also somewhat in proximity of each other. So this is a Ravel-like organization, and we check the clustering of centromeres at the genomic periphery by performing a SNPA immunofluorescence staining using these yeasts. And what we found is that the centromere cluster at the genomic periphery, at the nuclear periphery, while the genome is organized like a Ravel-like structure. So this is good, because we were assuming that the last common ancestor had a clustered centromere, And now we saw that both Candida tropicalis and Candida albicans, they have a rabble-like structure in their genome. And that probably indicates that the last common ancestor also had a clustered and rabble-like organization of the centromeres. Now, having identified this clustered rabble-like organization in the last common ancestor, we wanted to test if the proximity near the centromeres allowed the chromosomes to undergo translocation specifically near the centromeres or not. Now, enabled with this chromosome level assembly, we mapped all the synteny breakpoint between these two species. And if you try to find out where the chromosomes broke, you will see that the centromere proximal regions contain the most of the synteny breaks. As you can see here, these are the chromosomes of Candida tropicalis. This black arrowheads points towards the centromere, and you map the synteny breakpoint near the centromeres, but the chromosomal arms are mostly intact, as you can see in this figure. So we quantified and we found that the interchromosomal synteny breakpoint density, which is closer to the centromere, but as you go away from the centromere, the propensity of the centromere, propensity of the interchromosomal synteny breakpoint reduces strongly. So we also tested to find out whether the two regions which are proximal in Candida tropicalis-like state, does it converge in one chromosome in Candida albicans-like state if they went through the translocation. So we found multiple locations, as you can see here, centromere proximal genomic regions from two different chromosomes of Candida tropicalis are now located in the genomic proximity in the Candida albicans chromosomes. So that tells you that these two regions went through translocation and led to formation of this chromosome, which is the part of Candida albicans genome now. So having identified these proximity guided translocations, we were asking, can we find out whether the last common ancestor had inverted repeat associated structure or not? Because the centromeres are proximal to each other, but if you don't have DNA sequence identity, then it cannot undergo translocation. So we wanted to find out if there are other species in this complex which are also inverted repeat associated, and from that data, can we identify the last common ancestor between Candida tropicalis, which is here, and Candida albicans, the last common ancestor between these two species, that this hypothetical point here, can we say that this last common ancestor also had inverted repeat associated centromeres? So using synteny and presence of inverted repeat associated structures, we could predict the centromeres in multiple Candida species, including Candida parapsilosis, Candida bisphanathi, and Candida sojae. And we found inverted repeat associated centromeres in all three species. So this was kind of interesting for us, that we could say that the species which has diverged before the divergence between Candida tropicalis and Candida albicans also had inverted repeat associated structures. So that probably indicate the last common ancestor between Candida tropicalis and Candida albicans might have inverted repeat associated homogenized centromere. While we are doing this work, there was another report from another group here, Ola et al. They published the centromeres of Candida parapsilosis, and as it happened that our predictions, what we predicted for Candida tropicalis, Candida parapsilosis centromeres, they validated the centromere by ChIP-seq analysis. So this is well and good. We have the centromeres in last common ancestor that is clustered and inverted repeat associated homogenized centromeres. We wanted to see what kind of homogenization is there across this species complex. So this is what we are looking at, is a synteny dot plot. And this means is that if you find a dot in this matrix, if x-axis and y-axis elements are similar. So here you have the concatenated sequence of Candida parapsilosis, sojae, Candida tropicalis, Candida bisphanathi, and so on and so forth. So if you pick up the spot here in the matrix, you know that the sequences in the centromeres of one species has homology with another species. And we found that Candida sojae, Candida tropicalis, Candida biswanathi centromeres, as you can see here, they're conserved, homogenized, inverted repeat associated structures. But as you can see here, Candida albicans centromeres are almost unique centromeres. So are the centromeres of Candida dubliniensis. So we compared the genomic sequence identity across the centromeres within the species. And if we compare between the species, what we see that the centromeres are indeed homogenized inverted repeat associated regions across the species. As you can see at the species pair, we compare the sequence identity across the species pairs and also within the species. So having identified these homogenized inverted repeat associated structures in closely related Candida species complex, and with the observation that the centromeres in Candida tropicalis has a rabble-like structure in which the centromeres are organized at the periphery of the nucleus, we proposed this model that the centromeres in the last common ancestor had clustered inverted repeat associated homogenized centromere that went through translocation, and during the process they might have lost the inverted repeat associated sequence to generate the new evolutionary new centromeres, which are presented in the Candida albicans like state today. But the important point is that while we would expect that during the centromere evolution the genomic organization might change, that is not the case here. As you can see, both the species, Candida albicans and Candida tropicalis, both of them retains the rabble-like structures. So it tells you that the fungal genome organization is one of the, this is centromere clustering is one of the main feature of the genome organization in fungi. So with that, I would like to conclude my talk and thank all the members of MML, especially Kostu. This is Radha, Bhagya, Yao, Dr. Amartya Sanyal from NTU Singapore, whose lab, who helped us with the 3C-seq and the analysis part. And we also got some help from Geraldine and her group from UCD Dublin. And we got funding support from CSIR and Jane CSIR. This is a picture which is taken in December 2019. As you can see, my lab mates are all smiling and happy, and nobody's wearing a mask. So thank you, guys.
That was a fantastic talk, and I think super interesting questions. Oh, we already have a question in the Q&A. Okay, cool. So Janet Young says, nice talk. I agree. What's going on with chromosome 4? She asks.
Chromosome 4, you mean this one? So interestingly, if you look at this chromosome and chromosome 6, you would see that there is no break at chromosome 6 and chromosome 4. But this tells you this. So this is a little bit complicated, and I did not explain you properly. Maybe we can catch up later and explain you properly. But if I may quickly answer your question, is that this is not an interchromosomal synteny break, but these are intrachromosomal synteny break. That means there is a translocation within a chromosome, or you can say this is an inversion.
Oh, cool. So she has oh, okay, cool. You're getting a bunch of questions. So she has one follow-up question, which I think is interesting as well. So they say they think that in your C3 map, it looked like it might have 2 centromeres.
In this one?
I think so.
Are you pointing about this one?
That might be, because that looks
Okay,
very
so I'm sorry. So if you're asking about this one, this is actually happening because in Candida tropicalis genome, you have a translocation between chromosome 1 and chromosome 4. And because of this, you see this necktie pattern here.
cool.
And we validated this by doing chromoblot analysis, that two homologs of chromosome 1 and chromosome 4 are present in this configuration.
We have another question from Nick Sapp. He says, great talk. So with the loss of the inverted repeats with evolution, what defines the formation of centromeres in the absence of the inverted repeat?
I didn't get the question.
So he's asking, so with the loss of the inverted repeats with evolution, what defines then the formation of centromeres in the absence of the inverted repeat?
Great question. I mean, I have no idea, but you see, so when this translocation happened, the regions, so this is what we're looking at, two extant species in today's world, and this happened during, I don't know when, probably during the speciation, which is 39 million years ago. And then if you look at these translocations, what you find is that the centromere proximal regions in Candida tropicalis are merging together in the Candida albicans genome. But this line here is also the centromere in Candida albicans. So you see, the centromeres are forming near the region where they merged. I don't know what it means, but it may mean that so if you look at these cartoons, the centromeres are proximal to each other, and they're supposed to be a Saint-Pierre gradient near the cluster, which is explained as Saint-Pierre cloud. So if there is a loss of centromere, the preference of the regions which are physically proximal to the native centromere, they have the higher chance to form a Saint-Pierre region, which will next propagate it as newly evolved centromere. This is my best guess, but we don't have any data to support that.
Oh, thank you so much. I think we're out of time for questions, but if you have more, feel free to join us in the coffee chat, like I said, right in the chat, and we will upgrade you to a panelist at the very end of the seminar. Thank you so much for that wonderful talk and fantastic work.